large uptake, so that's the reason why it's higher. But what you definitely see is an increase in the signal with time. So that's the increase of CO2 in the atmosphere. Looking just at this portion of the record and blowing that up, now those data are these red dots right here. You can see that annual seasonal cycle I was mentioning. But also what's plotted on here are data from an oceanographic station, Station Aloha, in the Pacific Ocean. And what you see here in blue is the seawater PCO2, the partial pressure of carbon dioxide, that is rising. And what's really notable here is that it's rising at the same rate, with a similar slope as the atmospheric CO2. And then what's shown in this bottom panel here is pH measurements that were also taken from the seawater, and you see the decline in pH, which is exactly what you would predict if you're putting CO2 in the water. And I'll explain that a little bit. So where is the CO2 coming from? Well, here is a graphic that shows us that. So 90% of it comes from combustion of fossil fuels. So that's the big contributor. A smaller amount comes from deforestation. And so why does deforestation, okay, let's see, don't show this message again. All right, sorry for that. Deforestation changes the CO2 because you're taking away photosynthetic trees, and so they're no longer pulling the CO2 out of the atmosphere. All right, so we have an increase in the atmospheric CO2. Where does it go? Almost half of it stays in the atmosphere, and that's what the Keeling curve has measured. About a quarter of it goes into the photosynthesizing organisms from terrestrial plants like trees and grasses and shrubs, and then the other quarter to, sometimes you see this as high as 30%, actually goes into the ocean. All right, so the oceans are an important sink for the CO2 signal. And so if the oceans absorb about 25 to 30% of the carbon dioxide emitted, what happens to that? Well, you hear a lot about climate change in the temperature, and Ian referred to this. The other effect is that CO2 goes into the surface of the ocean and dissolves across the air-sea interface, and so then becomes dissolved in seawater. Now, I'm not going to go into the chemistry today in the interest of time, but basically what happens is that creates an acidic reaction that lowers pH. And the other thing that it does is it decreases a really important other chemical building block, which is carbonate. The carbonate ion decreases carbonate availability in the ocean. And so let's first talk about pH, one thing at a time. So pH, many people don't necessarily know, but pH is on a log scale, like the Richter scale for earthquakes. And so a change in pH of 1 unit means a 10-fold change in the acidity. And so here's seawater at around 8. And so when CO2 goes into seawater, it's still basic, but it's making things more acidic. And so the two things to remember here is that small changes in pH means much larger changes in the acidity. And sorry. And the second thing is that when the seawater changes, it's in the acidification direction. That doesn't mean that the seawater itself will be acidic, and that's very unlikely to happen, even in the long term. But it is truly acidification, just like when you have temperatures at 10 degrees and then it warms up to maybe above freezing. It's still cold, but we call it warming. All right, so now here's another graphic. And I apologize for all the graphics here, but it is the best way to really show the data. So here we have time on the bottom axis and then pH, and so you can see that as the CO2 in seawater has risen with time, the commensurate drop in the pH as well as the drop in carbonate ion. And that's just involved in the reactions once the CO2 gets into the seawater. And so what we see at the time horizon of the year 2000 is that we've dropped the pH 0.1 unit, which doesn't maybe sound like very much, but that's a 30% increase in the acidity and a 16% increase or decrease in the carbonate ion. Now, as we project this out into the future, it gets worse, and that's the unnerving thing about CO2 is that it's not a linear reaction. As time goes by, because of the great reservoir of the oceans and how effective it is at absorbing atmospheric CO2, and as those waters travel through the ocean and upwell and become more and more apparent, then these increases in acidity become quite large, as well as the decrease in carbonate. All right, so to sum it up, ocean acidification, CO2 added to seawater changes the water chemistry, reducing the pH and the carbonate levels in the ocean. So now let's go to the oyster for the second half of the story. So we understand the atmospheric story from the fact that Keeling actually had those data and allowed us to really see that atmospheric CO2 was changing. Oysters have actually been a poster child for helping us understand the impacts of OA. And it first started out with just the shellfish growers really noticing that something was happening. And here's an article from the Seattle Times that was published, I think, back in 2005. People started noticing that the natural set wasn't happening. They failed to reproduce. And so the problem is that, you know, as it says here, everyone knows nature can be fickle, but the problem is it happened again and again and again and again. And then you had folks down at a shellfish hatchery in Oregon called Whiskey Creek Shellfish Hatchery who started doing some observations and experiments with scientists from the Oregon State University, Alan Barton being one of them, Burke Hales and George Waldbusser. And here's some of their data, which was published in 2012, that shows this value here, which I haven't defined yet, but that's something called the omega. It stands for the saturation level of one of the calcium carbonate biominerals. In this case, it's aragonite, which is what oysters actually use for their shells. So it's basically telling you how soluble aragonite is. And if it's above 1, that means that the shell can be laid down. If it's below 1, that means that it would start to dissolve and so therefore couldn't get laid down. But you can see that even when you're above 1, there's a negative effect on both the production and the growth. And to see this visually, here's pictures of oyster larvae where aragonite saturation is at over 1, at 1.6, and here it is when it's below 1. So that, you know, picture tells 1,000 words. So clearly this is having a demonstrable effect on oyster larvae. And so here's really to sum up what we know about ocean acidification. We know that the atmospheric CO2 has increased from human activity. We know that the oceans absorb CO2. We know that the chemistry changes and that reduces pH and carbonate levels. This has been measured. And we also know that ocean acidification, this effect, whether it's on carbonate or on pH, reduces growth and survivorship in some organisms. And it's this last one that the scientific community is really working on better understanding. What organisms and how is this effect happening? And obviously on organisms that have calcium carbonate shells, we know dissolution is a factor. But on other organisms, how does it affect their growth and survivorship? And so one of the things that I really think is important to realize about ocean acidification is that it's a global condition, but with very local effects. And so here's my underpinning for that. Here I showed you the PCO2 in seawater, the concentration of CO2 in seawater from the Mauna Loa station in Hawaii. That's the golden time series. But here is that same time series in Bermuda and also in the Canary Islands. And the black is the atmospheric CO2. And so you see that all these ocean basins, whether you're in Bermuda, Canary Islands, or Hawaii, are showing the same effect, not only for the CO2, but also for the drop in pH and the drop in carbonate. And this is from the IPCC. But here, I think this picture is also really stunning. Local effects. So this person right here in the pink jacket was the former governor of Washington State, Christine Gregmar. The gentleman in the jacket is Bill Dewey, who works for Taylor Shellfish. And so he was one of the shellfish growers from the Pacific Northwest that started letting their decision makers know that things weren't working the same. And so here's a very local effect on that beach. And in order to, let me actually back up. So what the governor did is she convened a blue ribbon panel of experts, not only scientists, but policymakers, agency leads, tribal chairs, NGOs, organizations, and got them to discuss what do we know, what do we need to know, and what should we do at this point in time. And what's really important about ocean acidification is because these effects are very local, is that one needs to understand their system. And so here's a picture from our panel report, which is available online. I think you can just Google that and it'll get you there. But in the Pacific Northwest, we have processes like upwelling, we have river inputs, we have all sorts of things going on in the physical, chemical, and biological reactions. And it was important to consider this in the context of that. And so here's some data that really illustrate that point. So what we have here, this is a picture of depth on this axis and then distance from the shoreline, which is this gray thing. And this is West Coast data on the left-hand side and East Coast data on the right-hand side. West Coast has very narrow continental shelves. East Coast has a very wide continental shelf. And so then what's shown in the three panels going downwards are salinity, oxygen, and this saturation state of aragonite. And what you see is where's the low aragonite? It's this blue colored water. And you see that on the West Coast, the blue colored water is at depth, but then as you get close to the coast, it follows the shoreline upwards. And you see that signal in the low oxygen and in the high salinity. And you don't see that phenomenon happening so much on the East Coast. And so what is happening on the West Coast is this process called upwelling. And here's a little picture of it, but when you have equatorward winds, that causes, due to the Coriolis force, the offshore water displacement, and then that brings the deep waters up. And so we're bringing the high CO2 and low pH water to the surface. And so upwelling has always happened, of course, and corrosive waters, low pH waters, have always been in existence. But the problem is, from the addition of the atmospheric CO2, we're adding more, so it's more corrosive. And folks studying this off the Pacific Northwest have found that you get corrosive waters instead of 11% of the time, which you had during the pre-industrial era, you now have it 33% of the time. So you're increasing the frequency of organisms encountering this stress. And other processes like the respiration at depth also contribute to CO2, so how productive an area is will have an influence on its natural amount of CO2. But then the question has to be asked, are local stressors contributing also? Things that stimulate production, like nutrient additions, or things that load carbon into the receiving waters, as well as atmospheric end members. And these are questions that are being asked now and really haven't been studied, and it's going to vary system by system because it's sort of it's a scaling issue in terms of whether the inputs are large enough to have an effect in the receiving waters. So we've talked a lot about the water and the physics and the chemistry. What about the biology? So as we mentioned, the effect on the carbonate is really important for organisms that have calcium carbonate shells. And so we spent some time talking about oysters, but here's a creature that you may or may not be familiar with. Its scientific term is a pteropod. People call them sea butterflies. They're a member of the plankton. They are quite small, but here's what happens to the pteropod when it's exposed to these corrosive waters. The shell starts to dissolve, and this was recently published by Nina Bednarsik with NOAA in the Royal Academy proceedings. And what she found is that under current levels today, some of these pteropods are dissolving. Pteropods are important members of the food web, so this could have repercussions to organisms like salmon, and this is another thing that needs to be better studied. But as I mentioned, what are other impacts, such as the nervous system of fish, the increase in toxicity of harmful algal blooms, and the potential loss of water quality benefits because we have fewer shellfish? So these are all things that have been studied to some extent in some places, localized areas, and we need to understand this. People are also putting some effort into wondering what responses could help us meet the challenge locally. Because seagrasses and kelps are photosynthetically, they take up that CO2. So people are seeing whether on a local scale could we mitigate some of these effects if we have rafts of floating algae or kelp. And then also there's the fact that some species may be able to adapt, and we just really don't know that. The sobering thing that I find with the issue of ocean acidification is the rate of change is rapid. This is showing CO2 concentrations from pre-industrial through actually not even current. This only shows through 2000. And here we were down at about 370. Now the chart goes out to 2014, and we're already off the chart. We're already up here at 400. So the problem is that, as you know, biological systems have evolved, things change, they adapt. But the problem is if that happens too quickly, some organisms may not be able to adapt. And so the rate of change is really important. So this leaves us with a lot of questions, and this is really where the scientific community needs to step up the intensity. What determines localized nearshore CO2 levels? How can they be measured better? So some people are looking at better sensor technologies. What management practices could reduce the effects of local OA? How fast and how far can species acclimate and adapt? What are the crucial tipping points? And which ecosystems are more or less resilient? You know, when you think of an upwelling system, those organisms have been exposed to corrosive water, so maybe they're used to it and they can adapt. Or maybe they'll be more susceptible because at some point you actually reach a threshold. So anyways, that's the basic nutshell of what ocean acidification is about and some of the status and future directions. Thank you.
Thank you very much, Jen. And if you could stop showing your screen, Christina, we'll switch the presenter to Paul Hirschberger. I apologize for the sound quality. I think it was, there was some feedback, Jan, I think maybe from your microphone. It wasn't anything that ITEP could control. But anyway, hopefully,
Sorry.
that's okay. Hopefully Paul Hirschberger will come in loud and clear. And Paul, are you on?
Yes, I'm on.
Okay. I will go ahead and introduce you while the screen gets switched over to you. Paul Hirschberger is the station leader at the U.S. Geological Survey, Marrowstone Marine Field Station, where he leads a team of federal scientists, students, and postdocs investigating the effects of infectious and parasitic diseases on wild marine and anadromous fishes. He is the member of the affiliate faculty at the School of Aquatic and Fisheries Sciences at the University of Washington and he is currently the president of the Fish Health section of the American Fisheries Society. And let's see, uh, I'm on my screen, I just see a big black screen, so hopefully your presentation will come up momentarily.
Okay, you see it now?
Yes, it looks good now. Thank you.
Okay, well, I'd like to thank the organizers and also thank everybody who's in the audience for coming out today. As was introduced, I work at the Marrowstone Marine Station. We're a USGS lab on Marrowstone Island, which is near Port Townsend in Washington State. And the real focus of our facility is trying to understand disease processes and how those processes impact wild marine fishes. What I wanted to talk about today really was the effect of climate change on these disease processes, and in particular, the direct effects of climate change, which are most commonly thought of as temperature effects. And I wanted to touch briefly at the end on indirect effects of climate change, which are really, in my view, changes in assemblages, whether those be fish assemblages or whether those be invertebrate assemblages, and how those changes in assemblages can impact disease processes as well. So I wanted to start with some of the really good historical data, speaking of direct effects of temperature. This work was published by Dr. John Fryer and his colleagues at Oregon State back in the 1970s, and they looked at several pathogens of fishes and the effects of temperature on those pathogens. And the first one here is Flavobacterium columnari, which causes columnaris disease, and this is in coho salmon. And here we have cumulative mortality on the vertical axis and temperature on the horizontal axis. And as you can see, as the temperature goes up, more and more fish get sick and die. Up to about 17 degrees here, now all the fish were dead in this particular challenge. This isn't the case with all pathogens and all diseases. Here we have infectious hematopoietic necrosis virus, or IHN, and kokanee as the host. And in this particular case, cooler temperatures are more damaging to the host, where we see 60% mortality at about 12 degrees, decreasing to about 20% mortality as the temperature increases. And other pathogens have the sort of sweet spot. This is Rhinobacterium salmoninarum, causes bacterial kidney disease in coho. And here we see the sort of sweet spot at between 7 and 12 degrees, where we see a lot of mortality, and the mortality drops off at cooler temperatures and again at warmer temperatures. So there's not a real blanket statement that can be said about temperature and disease. Each host pathogen relationship is individual and unique. But what I wanted to get across in this talk is this sort of conceptual model of disease. And really, disease is caused by an interaction of these three components: the environment, the host, and the pathogen. And under normal situations, these three components coexist in harmony without any disease occurring. But when perturbations happen, for example, climate change or other perturbations, these three components come out of alignment, and you get this area of overlap here in the middle, and this is where we get disease occurring. And what I wanted to do was go through, give an example of each of these different components and how perturbations to either the host pathogen or the environment can cause disease. So the first example involves climate change influencing host susceptibility. So in this case, we have a host shift caused by some perturbation, perhaps climate change, and we get this area of overlap here where disease is occurring. And I threw this picture in here because I knew it was lunchtime, and probably many of you are eating your lunch now looking at a sick fish, so I apologize. But this is VHS disease in Pacific herring. It's classic signs here. We have hemorrhaging in the mouth, the eyes, the bases of the fins. And this particular disease is endemic in this area in the Pacific Northwest, and it occurs in herring, and it kills herring quite quickly, and we see massive epizootics in herring and other fish throughout this area. What we're beginning to realize now is that the disease is influenced very much by temperature. Here is some work that we did looking at the effects of temperature on mortality of herring that were exposed at 3 different temperatures. Here we had a cool temperature, and we had about 80% mortality after about 3 weeks. At ambient, in this case, it was about 11 degrees centigrade. The cumulative mortality dropped about 40%. And at the warmer temperature, just shy of 15 degrees, we only had about 10% mortality. So this particular host pathogen model is very similar to what I just showed with the IHN and kokanee example that Dr. Fryer described in the 1970s. But in addition to mortality, temperature also impacts other dynamics of the disease. For example, viral shedding, the amount of virus that's shed by each of these fish after they've been exposed. So here we have the time of exposure. They were exposed here on day 0. It's days post-exposure on the horizontal, and the amount of virus that was shed after exposure. So at the cold temperature, we had a peak in viral shedding at 10 days. Warmer temperature, the peak occurred earlier, several days earlier, and the peak was of lower magnitude than we saw at the cooler temperature. And at the warmer temperature, again, an earlier shift in the peak, and the peak had a lower magnitude. Okay, so after 4 weeks, when the study was over, these fish, some of the fish were still carrying virus. So this is the prevalence of the carriers in the population at each of the temperatures. And at the cool temperature, we had 23 survivors, and about 70% of those fish were still carrying the virus. At ambient, 51 survivors, and only about 12% of those fish were carrying virus. And many survivors in the warm temperature, and none of those survivors after 4 weeks were carrying the virus. So temperature is influencing not only mortality, but also the persistence of the virus in these fish. Not only the prevalence of fish that were carrying the virus, but the amount of virus that was being carried in these fish. So here we have the total amount of virus in the tissues of these fish after a month after exposure. And in the cool temperature, on average, the positive fish had about 100,000 virus particles per gram of tissue, extremely high viral titers. The positive fish in the ambient group, about a 10-fold step less in viral titers, so here about 10,000 virus particles per gram of tissue. And again, in the warm group, none of the fish that survived had any virus that was recoverable. So what is causing this temperature relationship in this particular host pathogen example? And in this case, it really boils down to the immune response. And these next 3 graphs that I'll show you are a reflection of the MX response. MX is a herring immune response gene, and really what the MX response indicates is how well the fish is producing an antiviral response. So if we have a large number here in the fold increase of MX, we have a large antiviral response. And here we can see at the cool temperature, we had a large antiviral response at 10 days post-exposure. In the ambient group, the peak in the antiviral response occurred several days earlier, at 6 days. And at the warm temperature, the antiviral response occurred as early as 2 days after exposure. So what this is indicating is that the cool temperature, there was immunosuppression in these fish. So it's an example of temperature influencing the susceptibility of a fish as a result of immunosuppression. Okay, the other thing that we can have in a direct response to temperature is a shift in pathogen virulence. So in this case, we'd have this area of overlap here where we have disease occurring. And the example that I'm going to use for this is it's a parasite called Ichthyophonus. And the host system is the Yukon River and Chinook salmon that were returning to the Yukon River. So here on the right, this is on the left, this is an Eskimo lady at the mouth of the Yukon cutting king salmon with her ulu. At the right is a map of Alaska. Here we have the mouth of the Yukon River and a village called Amonic. And the river cuts straight across Alaska up into the Yukon Territory and the fish go about 1,700 miles up to the town of Whitehorse in the Yukon Territory. About halfway up the river, a large proportion of the fish cut off into the Tanana River here. And of the fish that turn into the Tanana River system, 90% of them spawn in these 2 streams here, the Chena and the Salcha. And those will be important here in the next couple of figures. But what we did was we followed the fish as they were migrating from the mouth of the river all the way up to Whitehorse and all the way up into these spawning tributaries and followed not only the prevalence of infection, but also the intensity of the infection of Ichthyophonus. And here on the horizontal axis, we have river mile. River mile 24 is down at the mouth of the river, all the way up to river mile 1700, which is up at Whitehorse. On the vertical axis, we have prevalence of infection. So the fish came into the river infected. About 30% of the fish were infected. The white portion of the bars here are the low-level subclinical infections. These are fish that were infected but not yet diseased. The gray portion of the bars were the fish that were demonstrating clinical signs of disease. And what this figure is showing is that as the fish are migrating upriver, the total prevalence of infection is staying fairly similar until the fish get up to the top of the river. What is changing, though, is that the prevalence of the proportion of diseased fish is increasing about halfway up the river. Now, the majority of the fish that we sampled were not only infected, but they were also diseased. And again, both of these dropped when the fish got to the top of the river before they spawned. This is a little better demonstrated when we look at just the Tanana River system. Here we have the same sort of figure, infection prevalence, diseased fish in the gray, just infected fish in the white. And again, this is halfway up the river now, so most of the fish are diseased. And then as the fish turned into the spawning streams, the Chena and the Salcha, these infected and diseased fish vanished. They didn't get to the spawning grounds. So we lost 60% of the infected and diseased fish, and in all likelihood, they died from the disease. So the question is, what was causing this particular epizootic, and why did it show up? The leading hypothesis at this point was temperature. These are the river temperatures during the course of that epizootic in the late 1990s and early 2000s. The circles are the mean temperature, and the dots above and below are the maximum and minimum that were recorded during the period. And I have this line cut through here at 15 degrees because this seems to be the magic temperature when it comes to Ichthyophonus and salmonids. And I'll show you those data here in this next figure. These are data from Dr. Okamoto that were published in 1985. And again, it's mortality with temperature here. And these are rainbow trout that were infected with Ichthyophonus at different temperatures and maintained at these temperatures. And at 10 degrees, he had about 10% mortality. But as soon as the temperature jumped up to 15 degrees, he had 100% mortality, and that persisted at 20 degrees as well. So 15 degrees seems to be the sweet spot for Ichthyophonus in converting it from a low-level infection to overt disease and mortality. And again, those were the temperatures that we were seeing in the Yukon. We can see, what's sort of causing this, and we believe it was an artifact of changes in pathogen virulence. And you can see this sort of qualitatively if you look at Ichthyophonus in a culture tube. This is the parasite here. This is some stimulation of hyphal growth. You can see the parasite protoplasm sort of coming out into this hyphal tube at 5 degrees. At 10 degrees, growth is a lot more. It turns into it looks like a head of cauliflower at 15 degrees, and at 20 degrees, it really takes off. So the parasite grows much faster at warmer temperatures. You can see this more quantitatively if you look at parasite spore production at temperature. At 5 degrees, very little spore production increased, and spore production increased with temperature up to, after a week, one spore turned into about 100 spores at 20 degrees. Okay. The last example that I wanted to give was an example of climate change and environmental variables. And no longer am I talking about direct effects of temperature, but these effects are indirect effects, and they're much more difficult to quantify. So here we would have a shift in the environment caused by climate change or some other perturbation that would create this disease situation here. And the first example I want to talk about is changes in ocean circulation patterns that are likely to influence fish assemblages. And we see this already with these changes in El Niños and La Niña cycles when we see, especially in El Niños, we see warm water fish species occurring in the Pacific Northwest from the south and temporarily residualizing in this area. When they come up from the south, they bring with them their own suite of pathogens that our native fishes in the Northwest likely have never been exposed to, and they become susceptible to. Conversely, when these fish come up from the south, they get exposed to endemic pathogens that are in the Northwest that they've never been exposed to, and they can get sick and die from those because they've never been exposed, they have no resistance to. And this newspaper article here is an example of that. This was a case of VHS virus, the same virus that I described in herring, killing sardines in British Columbia. And this is quite common during warm water periods. These sardines move up and semi-residualize in the northwest, including the Washington coast of British Columbia. And then as those warm water refugia break down, these sardines are trapped in embayments and fjords where the warm water still subsists. And eventually, those areas break down as well. And as I just described, as the temperature decreases, the susceptibility of these fishes to VHS increases. So we see these epizootics in sardines, and very likely these epizootics during the cold water events after they've come up here. And very likely these epizootics then spill over to our native species like Pacific herring. Okay, the other the other effect is changes in zooplankton assemblages. And I just threw these two pictures up here. This is a copepod and a euphalsid. And we get these different changes in assemblages during warm water periods and cold water periods that we already are able to see. And in addition to just providing basic nutrition for fishes, these invertebrates are really important because they serve as intermediate hosts for a suite of pathogens that affect fishes. Nematodes, digenean trematodes, as well as myxozoans are well known to go through these intermediate hosts. And when we have switches in the assemblages, it very likely is going to impact the pathogens that the fish are exposed to. And I'll give you an example here. Again, this is Ichthyophonus and Pacific herring. These little the black spots on the flank of this fish are ulcers where the parasite has migrated from the inside of the fish and ruptured through the skin, and it's released into the water. We're now referring to Ichthyophonus as the most significant pathogen of wild marine fishes in the world. And the reason we say that is because Ichthyophonus has a precedence of massive, large-scale epizootics that have occurred over the last century in wild fishes throughout the world, especially in clupeids like Pacific herring and shad. We've tried for several years now to try to infect herring with Ichthyophonus, and the only way we can get them infected is by injecting them with the parasite, which is really unusual because in the wild right now, the mean prevalence of Ichthyophonus in the Northeast Pacific in adult herring populations is anywhere from 30 to 50%. So there seems to be some mechanism that we're missing when we're trying to get our herring infected. We've tried feeding them Ichthyophonus in their food. We've tried intubating the parasite directly into their stomach. We've tried cohabitating infected individuals with uninfected individuals. And in each of these processes, we've been unable to establish infections. At this point, the most parsimonious explanation accounting for the transmission of Ichthyophonus into Pacific herring involves an intermediate host, such as one of these that I just described. And very likely, these epizootics that we see crop up unexpectedly and very rapidly are very likely the result of changes in zooplankton assemblages that are carrying that are serving as carriers for this intermediate host. Okay, so to summarize, climate change can influence disease processes either directly by direct effects of temperature or indirectly by changing ocean circulation patterns and other processes that affect both fish assemblages and invertebrate assemblages. This is a photograph of our field station on the Marrowstone Island. And as you might suspect, we're keenly interested in the effects of climate change, especially as Ian mentioned, on changes in sea level rise. And this is an event that we had, a storm event that we had several years ago, where we actually had water up to our gates. So thank you very much for your time.
Okay, thank you very much, Paul. If you could stop showing your screen, and Christina will change it over to Paul Williams. And while that is happening, I will introduce Paul. Paul has been a shellfish biologist at the Suquamish tribe since 1992. He has been involved in every aspect of achieving and implementing state and tribal co-management of the many shellfish resources throughout the Puget Sound. More recently, he has become involved with efforts to protect those resources through developing new ways to monitor zooplankton and compiling and disseminating educational materials on ocean sciences for K-12 teachers. He's just going to very briefly talk about a few of the things he's doing, and then we'll take the last few minutes to address a few of the questions that have been posted. And with that, Paul, it's over to you.
You hear me now?
Yes, I can.
Can people hear me?
Yes.
Okay, great. So thanks for organizing this. I think it's a wonderful opportunity for all of us to get some of the science from the scientists. And now, from you know, as she mentioned, I'm a tribal biologist, and a lot of folks have been working on solutions to ocean acidification and how to mitigate it. But we're thinking also along the lines of who is going to be addressing ocean acidification, and recognizing that a lot of behaviors really need to be changed. We are, in order to lessen some of the impacts humans are already having on Puget Sound, we're developing this ocean acidification curriculum collection, and it's not quite online yet, but you can filter by grade, filter type of activity, length of activity, and then we've got a lot of, yep, there we go. I'll just show you really briefly. We can, so this one is sorted for the carbon cycle game, and then you can go to the link on the carbon cycle game, and it shows you this cool link. And this nice little game for kids to learn about the carbon cycle. So that should be out shortly. We're really in need of help in terms of finding materials, and so if you have anything you would like to share, any materials, and this will be promoted to teachers, and you can send those materials to oacc@suquamish.nsn.us. We'll try to get this out probably within a few months. We've also been doing a lot of outreach, and this is a booth we set up at the paddle to Quinault. And this summer for the tribal canoe journey, what we'd like to do is, this takes a lot of time to set up a booth. So we're going to have just a string of posters on different topics, very simple, and we'll string that up at each stop along the journey to Bella Bella, and then we'll give that collection to the local school as we go. And so we're trying to develop those posters now, and if anybody wants to help out by helping in that process of development of the posters, please let me know. And the third thing, I don't have any slides on this, but we've recognized that some of the ocean acidification is really quite a new problem, and we don't have the tools to really study some of the species that are impacted the most, like zooplankton. There's several folks working on pH monitors, but we're looking at using the technology that's been developed in cell phones to repurpose that for imaging zooplankton. So the microcomputers that have are developed, they're very inexpensive now. We'd like to put that with imaging devices, line scan camera, or just a regular matrix camera. Put those underwater in a watertight box attached to a computer that could then identify what is in the image using techniques that have been developed for facial recognition. We have a few people working on this right now, a student from the bioengineering lab at the University of Washington, and also somebody in oceanography. But I found that it's really hard to break through those institutional silos to get folks to work together from different disciplines. And so those are kind of two of the initiatives we're working on, and I'd welcome to people to talk about it offline or later on, and I'll end now to provide folks with an opportunity to ask questions of all the wonderful speakers we've had. Thank you.
Okay, thank you, Paul. We do have a few questions that came in. First, about ocean acidification. So Jan, I think this is directed towards you. Do we know enough to provide advice on what can be scale to mitigate for increases in acidification of marine waters? And then there's also a part two. Is anyone looking at climate-related changes in pH levels in freshwater bodies?
Yeah, so I hope the sound quality is better now. Let me know.
It's okay.
Okay. I might have had the microphone too close. I haven't done this before. Anyways, so to answer the first question, at this point in time, not with scientific justification. And so in the state of Washington, which is where a lot of my knowledge stems from, we are probably a few years away from that. We have models being developed, hydrodynamic models that have loading from the watershed as well as the atmospheric and oceanic drivers in there as well. And you know, while these are used commonly for looking at hypoxia, lack of oxygen, and nutrient loading, getting the carbon parameters in there is new research. And so that's ongoing work. Plus, we probably need some better data. But I think in 2-3 years, something like that, we're going to be able to make some judgments. As I mentioned in the talk, it's kind of a scaling issue. So what's true for one part of Puget Sound wouldn't necessarily be true for another part of Puget Sound or San Francisco Bay or Chesapeake Bay. And so that's something that's going to be locally investigated. With respect to freshwater pH, I was at the Ocean Sciences meeting last February in Hawaii, and there were colleagues from the University of Wisconsin-Madison, Jen Phillips, and I just looked up her abstract to be able to read to you. It says, a number of studies indicate that air-water equilibrium controls PCO2 in several Great Lakes at annual timescales or longer. Assuming this is the case across all lakes at present and into the future, pH will decline by 0.3 to 0.4 units through 2100. So then she went on to say in her presentation that they really didn't have the data set because the monitoring strategy wasn't set up in a way that was really conducive to that. So it does look like there can be a freshwater effect, but that is, to my knowledge, much less studied.
Okay, thank you. And then Paul Hirschberger, while you were speaking, a question came in, is water quality an issue?
Sorry, I think I'm unmuted now. For the most part, traditional water quality like pH and temperature are not direct issues, but from an indirect perspective, yes, it is. In that water quality, in terms of contaminants, certainly can impact susceptibility to certain pathogens.
Okay, thank you. And then there was a question for Ian. How long has VLM, I guess that's vertical land movement, been taken into account in climate change impact reports?
That's a good question. I mean, it shows up, it's a consideration that is definitely included in some assessments. I mean, in a way, you can make an argument that sea level rise assessments are sort of a new thing. It does show up in NOAA published a guidance document on how to do local sea level rise projections, and it shows up there. And so, it's a factor I think that has been taken into account previously. Certainly, it's nothing new in regards to people know it's a factor in relative sea level rise and have for some time. I think more the issue is having good data available on being able to characterize local rates of vertical land movement. Seems to be the big issue.
Okay, thank you. And I believe that's all the questions. I'm sure any of the presenters would welcome further questions, maybe via phone or email. So if any of you remaining on the webinar have any more questions, please feel free to contact the presenters. And at this point, we're going to end the webinar. I want to thank all the presenters for joining us and sharing all the great information. And I invite everyone to join us for our next webinar, which is next week, May 27th. Okay, with that, we'll close the webinar. Goodbye.